Muy buenos días y bienvenidos a este seminario virtual. Soy Miguel Urdaneta, profesor de los cursos de Microbiología Aplicada y Microbiología Industrial de la Universidad de Puerto Rico, recinto Río Piedras, y estaré participando como moderador en este evento. Es importante informarles que la actividad está siendo grabada en su totalidad. Para propiciar que la presentación fluya sin distracciones, solicitamos a la audiencia apagar sus cámaras y micrófonos. Esta actividad surge de la inquietud de nuestros estudiantes acerca de la procedencia y evolución del virus SARS CoV-2 que da origen al COVID-19. Para contestar algunas de nuestras interrogantes y compartir sus ideas sobre el tema, hemos invitado al doctor Massey. Adelante, doctor Massey. Sin más preámbulos, les dejo con el doctor Massey y su presentación del tema, la epidemiología genómica y futura evolución del SARS CoV-2. Adelante, doctor Massey.
Okay. Thank you, Miguel. You were breaking up a little bit there. Thank you, and thank you to everybody who's attending. So thank you to the students of industrial microbiology and everybody else who decided to come. My talk is entitled Genomic Epidemiology and Future Evolution of the Coronavirus SARS CoV-2. And I will be talking about the application of genomics and bioinformatic analysis to understanding this current pandemic and hopefully making some predictions as to its future progress. So we're in unusual times, as I'm sure you all realize, in terms of this pandemic. It's not the first pandemic that humanity has suffered from, but it's the one that spread the quickest, and this is due to globalization. Another interesting feature of this pandemic is the utility of science in combating the disease, and really the prominence of genomics and bioinformatic analysis in understanding the origin of the virus, its spread, and its future prognosis. So just a little bit, Miguel asked me to introduce myself a little bit to those of you who don't know. My name is Massey. I'm a professor of bioinformatics in the biology department at Rio Piedras. I have a PhD in comparative genomics of Candida albicans, which is a sometime pathogen. I specialize in molecular and genome evolution. So I'm an evolutionist in general. I'm interested in all sorts of evolutionary topics, but this is really my speciality. And throughout my career, I've focused on microbial genomics, essentially for partly pragmatic reasons. There's a lot more. And they tend to be somewhat quite a bit simpler than eukaryotic genomes. And so therefore, it's a little bit easier perhaps to see the underlying evolutionary forces at work on genome structure when one considers microbial genomes. And in my time, I've studied a variety of different pathogens, and it turns out that pathogen genomes are particularly interesting because of their conflict with the host, and so therefore they have a variety of unusual genome structures and adaptations. And so some of the genomes that I've studied include malaria, HIV, Helicobacter pylori, mycoplasmas, chlamydias, sea slug retroviruses, and nucleocytoplasmic large DNA viruses. So as I say, they're all interesting no matter what microbial lineage they're from, and they do actually show some similar dynamics because the conflict between the host and the pathogen has some general aspects that's applicable to all of these pathogens. Now, if any of you are interested in contacting me, then please feel free. And my email is this email here, so at the bottom, so stephen.massey@upr.edu. Okay, so what is COVID-19 and what is SARS-CoV-2 and what is the difference between the two? So COVID-19 stands for Coronavirus Disease 2019. Now, the name for the causative agent, which is a virus, is Severe Acute Respiratory Syndrome Coronavirus 2, SARS-CoV-2. And so this now is the commonly accepted name for the virus that's causing the COVID-19 pandemic. Now, SARS-CoV-2 is an enveloped positive sense, single-stranded RNA virus belonging to the Coronaviridae family. And the reason they're called coronaviruses is they have these spike proteins, see on the image on that, that gives it the appearance of a crown. And so it's commonly called coronavirus, but perhaps we shouldn't call it that because actually 20 to 30% of strains of the common cold are actually also coronaviruses, albeit from a different family of coronaviruses. So SARS-CoV-2 has a large genome for an RNA virus, one of the largest. It has a genome of 30 kilobase pairs, 30,000 base pairs, and it contains 15 genes. And this is actually important. I'll tell you why that is in a minute. Now, at the bottom is a scheme of the genome, and it's a linear genome. And some of you might be thinking, wait a minute, there's not 15 genes down there. And that's the reason. The reason for that is that the first two green open reading frames are what are called polyproteins, and they're cleaved subsequent to translation. Now, after those two green polyproteins, there's a protein called S protein, which is the spike protein that I just mentioned, and this is very important, as we're going to find out in a minute. So, it's a big topic right now, and obviously very important to know where this came from, because we want to be able to prevent this in the future, and we are vulnerable to future events of this type. Comparative genomics, this is really the power of genomics and analysis, is that comparative genomics has shown us that the likely source of SARS-CoV-2 is bats, and that is the ultimate source. And so this is because SARS-CoV-2 genome is 96% identical at nucleotide level to a coronavirus isolated from bats. And actually from this bat here, which is Rhinolophus finus, which is the intermediate horseshoe bat, which is found in caves in Southeast Asia. And so this represents what's called a zoonotic host jump. So when pathogens jump from one species to another, this is called a zoonosis event. And it happens from time to time. It's sort of a natural occurrence. But when we come into contact with animals that we don't normally come into contact with, then the chances of undergoing a zoonosis are obviously increased. And so this could be an explanation as to why SARS-CoV-2 is currently so deadly. It may be maladapted to the host, which is us. And so it prefers to live in bats. It's co-evolved with bats over a long period of time. And when it encounters a new host, they tend to have an unhappy initial relationship. So a well-adapted virus that depends on social interaction to spread should not kill the host quickly. And the reason is that this obviously would stop any further social interactions and potential opportunities to infect new hosts. So it's not in the interest of the virus to. So a critical portion of the SARS-CoV-2 spike protein shows 99% amino identity to a coronavirus recently isolated from pangolin. And so pangolins are actually sold in the markets, some markets in Southeast Asia, and they're considered a delicacy. And so this implies that a recombination event has occurred that has produced the current form of SARS-CoV-2, which is producing the deadly effects. So the recombination is predicted to enhance binding to the human virus receptor, which is called ACE2, and I'll talk about that a little bit in a minute. Now, the intermediate host is not known. It's important to find out if there was an intermediate host, because again, this tells us the likely origin of the virus, and that's obviously important to know. So the intermediate host could be human, and so sort of a basic sort of biology question is which predator eats both bats and pangolins, or perhaps some other unknown host? And there's not many predators of that nature, and probably humans fulfill that description the best. Actually, bats have very few natural predators, and the reason is, I go to the previous slide, because of those two large appendages on each side of its body called wings. And so they can fly and they can escape predators quite easily, and perhaps that's part of the reason for evolving flight in this mammalian lineage. And so bats don't have natural predators, so it's perhaps hard to understand how it could transfer to humans. Now, bat coronavirus has also been found in bat guano, and so this provides another potential route for interspecies transfer. And apparently, bat guano actually is used as a form of traditional medicine in some regions of Southeast Asia. And actually, other regions around the world, bat guano is a great fertilizer, so it's very rich in phosphorus, so it's widely used as a fertilizer. So this emphasizes that an additional danger of markets that sell a variety of wild animals is the possibility of combining viruses from several species, and those viruses undergoing a recombination event to produce a deadly recombinant. So there's an evolutionary theory called the trade that have jumped hosts should become less virulent. So that's good news for us, right? And so this is because if it's less virulent, then this would enhance the fitness of the virus, which means it would be likely to get fixed in the population of the virus. And the reason it would increase the virus is that being less virulent would promote its spread. So if you think of a bad dose of flu, for example, you're probably laid up in bed for a few days and you're not meeting anybody, you're not going out, hopefully, spreading that infection. Now, if you're only mildly sick with flu, you're probably going to continue working, you're going to interact with a lot of people, you might cough and sneeze a little bit, and you might unintentionally infect several people. And so, so the less virulent a pathogen is, often it's in its interests. Now, an exception to this are waterborne pathogens such as cholera, which can be highly virulent. That is, they can cause mortality rapidly without comprising their contagiousness, because once it's released into the water supply, it can spread and infect new persons relatively easily. However, in the case of SARS-CoV-2, there's a long period where the virus is asymptomatic but still transmissible before killing susceptible people. And by the way, it's not actually the virus that's killing people, it's an overreaction of the immune system, which is called a cytokine. So it's a massive release of cytokines that induce an inflammatory response, and essentially the immune system causes inflammation in the lungs can fill up with fluids, and people essentially drown in their own bodily fluids. And so very unpleasant, and but it doesn't do this to actually most people. And incidentally, this is the same story with the Spanish flu. The second wave of the Spanish flu, which was more deadly, caused these cytokine storms, and but this time actually in younger people as opposed to older people. And there was some thought that there was some immunity that older folks had actually experienced an epidemic of flu in the 1860s, I think it was, and so therefore they were perhaps more resistant to the Spanish flu, but it's not like that. If SARS-CoV-2 is asymptomatic, there's before it can suddenly trigger this this cytokine storm, then there's not much of a selection pressure to reduce virulence because it could have easily spread by that time. So what is the mechanism of infection? This is very important for understanding how to perhaps design antibodies, vaccines that is, drugs, and also as to why this has suddenly arisen in the fashion that it has. So SARS-CoV-2 spike protein binds to a human receptor protein called angiotensin converting enzyme 2, ACE2. And this protein is highly expressed in the respiratory tract, which is why SARS-CoV-2 is a respiratory virus. It attacks the respiratory tract. And so the normal binding partner of ACE2 is angiotensin, and it's a short peptide hormone, and that's cleaved by ACE2. Now, the receptor binding domain of spike protein, RBD, binds to a site on ACE2 actually distant from its active site. So in the scheme I've shown here, you can see the SARS-CoV-2 RBD, and it's binding away from, if you can see on the left, it's marked PD, and that stands for protease domain, and that's the normal binding site of angiotensin. So it binds in a atypical binding site on ACE2. Around the world, there are no, appears that there's no or very little population-specific variation in the region of ACE2 that binds to the spike protein. So this means that binding affinity of SARS-CoV-2 for the human host is unlikely to vary in different populations. So there's unlikely to be a genetic basis there. However, expression levels of ACE2 may vary between populations, but probably more important is segments within particular populations, which is age, people who are younger versus people who are older, people who have asthma, for example. Diabetes has been linked with it, obesity as well is strongly linked. So factors like that can affect expression level of ACE2, probably more than genetic differences between things. And of course, exposure to things such as contaminants in the air as well might affect expression levels. Pathogens, in general, pathogens and their hosts are engaged in what are called co-evolutionary arms races, and this is where they compete with each other for extended periods of evolutionary time, sometimes over millions of years. So on the right, I've shown an image that I think is probably familiar to all of you, which is a zebra on the African veldt, which is being chased by a lion, and the zebra has evolved mechanisms to avoid the lion, and one is speed, and the other one is a deceptive mechanism, which is the stripes, which allow it to blend into the background normally, but not here, and so perhaps why this is why being chased. The lion also has sharp claws to grab onto the zebra. It's evolved strength and agility to catch up with the zebra, and it does have its own form of camouflage, the brown color. Evolving adaptations over time, and this goes on and on and on over millions of years. So the zebra maybe evolves to be quicker, and then the lion then perhaps evolves into another species, maybe something like a cheetah, right? So, so this sort of process is also present in pathogens and their hosts at the molecular level, and so this is reflected in enhanced sequence variability in the pathogen virulence factors, such as spike protein, for instance. And and in the host receptors. And so the host receptors will evolve to avoid being recognized by a common pathogen. So consistent with this, the spike protein in SARS-CoV-2 is the most variable protein in the genome, and so this is telling us this is probably one of the most important proteins to be looking at in terms of virulence and interaction with the immune system. However, there is a lack of variation in the human receptor protein H2. So this, at least in the binding domain for spike corona, for SARS and SARS-CoV-2 coronavirus, and so this implies that this particular group of coronaviruses has not had an extended exposure to the human population. So, are telling us essentially that these viruses are essentially novel. Perhaps you could sort of stretch that interpretation and say that we're doing things that we haven't naturally done in the past, and we're coming into proximity with reservoir species that we haven't previously come into contact with. And I think there's a lesson there. So how do coronaviruses evade the immune system? So I showed you the picture of the zebra, which is trying to escape the predator, and then the predator actually also uses camouflage to deceive the prey. And so it's not a surprise that pathogens should also use all sorts of deceptive strategies, and coronaviruses are no different in that. And so coronaviruses actually dedicate a large part of their genome to deception, and so this is one of my interests, the role of deception at the level of molecular evolution, and I'm actually interested in deception actually in terms of evolutionary psychology and economics and things like that, but it's a topic for another day. So the coronaviruses do use all sorts of deceptive strategies. This is just a sample to give you a flavor of what's going on. So SARS-CoV-2 spike protein is covered in sugars, and these mask the virus from the immune system, so it's a form of camouflage. And on the right, I have a scheme, three-dimensional picture of the spike protein from SARS-CoV-2. A lot of very rapid and very high-quality research is coming out right now, and so it's quite incredible how quickly people are doing this. And so now this three-dimensional structure has sugars attached in color. And you can see that it covers the whole molecule, but there's some large gaps there. And it's actually not densely covered, and this actually means that there are multiple potential sites of vaccine design, which is good news. There are two proteins commonly found in coronaviruses called NSP3 and NSP4, and these mimic inclusion bodies, which induces the formation of replication organelles, which you can see on the left. And so this is where the viruses essentially hide and congregate and reproduce. And so these replication organelles act as a mask inside the cell, almost like a Trojan horse. And so it's a form of, again, of camouflage, and the form of, for that, a Q mimicry. And so coronavirus NSP16 adds caps to virus RNA, making them appear to the cell to look like cellular mRNA. And so this is a form of mimicry, and actually the equivalent in organismal biology is bait mimicry. And so coronaviruses also commonly possess an endonuclease activity that degrades their RNA after translation, and this reduces their footprint within the cell, and it avoids detection. So this is essentially a stealth strategy. And also there's a protein called PL-PRO, and this mimics cellular D-ubiquitinase, house USP7, and this allows it to inhibit immune detection. And there's actually several other mechanisms that they utilize. It's absolutely fascinating that they can pack in so many deceptive stratagems into such a tiny genome. It's truly miraculous, and it's telling us, and I'll talk about this in a minute, that it has a very strong conflict of interest with the host, which is the reason it has developed all of these deceptive strategies. Now, how is SARS-CoV-2 evolving? So SARS-CoV-2, as I mentioned, has a large genome, and our lab has, over the years, developed a hypothesis called proteomic constraint, and it proposes that replicational fidelity is related to genome size, which is equivalent to proteome size. And so large genomes have greater fidelity, and you can see on the graph on the right that there's a linear relationship between genome size and underlying mutation rate for a range of complete genomes, with a couple of exceptions. There's always exceptions in biology, right? So this means that SARS-CoV-2 should have a lower mutation rate than influenza, which has a smaller genome than SARS-CoV-2. And this would mean a lower rate of differentiation of SARS-CoV-2 than influenza. And this appears to be true. So it does appear that SARS-CoV-2 has a substitution rate, which is related to the mutation rate, lower than influenza. And the reason for this is that likely is that SARS-CoV-2 has a proofreading exonuclease protein, which corrects errors made by RNA polymerase during genome replication, while influenza actually lacks such a proofreading activity. And this is actually predicted by our proteomic constraint hypothesis, which is all very gratifying, but actually not a surprise to us. And actually, there's also good news here in that what it means is that a vaccine should be effective for a number of years, because the virus will not change as quickly as influenza, which requires a new vaccine every winter. So how is the virus adapting to humans, its new host? Because this can sort of reveal the future to us, really, and the future nature of the virus. And so SARS-CoV-2 has accumulated quite a few individual mutations since it emerged in November, December time in China. So it shows an excess of synonymous mutations, which are silent mutations. So if you look at the genetic code, you see some mutations wouldn't lead to a change of amino acids. So if you look at glycine in the bottom right, you can change the third codon position of glycine and it won't change the amino acid. That's a synonymous mutation. Now, non-synonymous mutation is where if you have a mutation, it will change to a new amino acid. So, and often that's the first and second codon positions result. If you mutate those, it results in a new amino acid change according to the genetic code. So synonymous mutations are a lot more common than non-synonymous, which means the non-synonymous are being removed from the population. So it's removing amino acid mutations, essentially. And that's what we call purifying selection. So it means that the virus is under quite strong selection. And so this means that we would expect it, therefore, to adapt its amino acid sequence to its new host. And this would mostly be expected to occur within virulence factors, for instance, that interact with ACE2, so that would be the spike protein, and also with virulence factors that interact with the immune system, both innate and adaptive. And I gave you a list on the previous slide of interact with both the adaptive and innate immune system. So, so a lot of potential adaptation going on. All of these will impact infectivity and transmission in one way or another. So, in addition, the silent mutations, the synonymous mutations, most people think that these don't do anything, but they actually affect translational efficiency. And so we would expect these to slowly mutate over time to match those of humans in order to optimize its translation in its new host, because the codon usage of humans is different from bats and actually other mammals. Another big question is, was SARS-CoV-2 engineered? And so the first thing to say is that technically it is straightforward to synthesize a modified virus genome, given current technology. However, designing a virus to have high virulence and contagiousness, for which the human population has little immunity, is not straightforward necessarily, and it might only be done with really sort of large resources and state-level resources. And so a lot of sort of things in the press were saying, well, this was deliberately released and somebody designed this. And I think if we're talking about engineering, we're perhaps, I don't think, I think it's sort of unlikely that anybody would want to necessarily to release something like this deliberately, unless it's a rogue state and they might not have the resources available. But certainly there is a precedent for engineering flu viruses, and in order to better understand them. So yes, these viruses do sometimes get engineered or altered within laboratory settings. I have to say that the intention is not to release these things. Now, none of the, so a number of publications have outlined the mutations that would actually improve the binding affinity of SARS spike protein for ACE2, and they could be transferred to SARS CoV-2. And so the information actually is out in the literature, but none of these were actually observed in SARS CoV-2. It does have a high affinity for ACE2, as I mentioned before, but this appears to be due to natural selection because of the unique combination of amino acid changes. And it has appeared to have occurred via recombination with pangolin spike protein. So it's now being reported that it's not likely that it's been engineered. Now, unfortunately, what hasn't really been discussed, and I think it's probably wise to discuss these, is that there is actually another change in SARS-CoV-2, a region that I'm not going to give too many details about, which is actually commonly known to a range of viruses, actually bacterial pathogens, actually also cancer, and this is also observed in SARS-CoV-2. And this particular region has actually already been engineered into flu strains to test whether it alters the infectivity of flu. So I guess the answer to the question of was SARS-CoV-2 engineered is we don't have the information to say that it wasn't right now. So we cannot say one way or the other. We don't have a signature that shows that it was, but we cannot actually say that it wasn't. Now, did it escape from a lab? So sequence analysis, bioinformatic analysis may reveal if there was intermediate host. So we need more samples from wild animals to really understand this recombination event, and we need to better understand the sequence, things like soil codon usage, in order to understand whether there was adaptation to an intermediate host. And so if there was an intermediate host, then it's not likely that it escaped from a laboratory. So this it probably evolved its present property inside a laboratory. Now, on the other hand, if it simply looks like a bat coronavirus and it evolved within bats and perhaps recombined with another bat coronavirus that had a sequence similar to pangolin, and maybe this is a common motif, then it would be it wouldn't be possible to falsify the hypothesis that it came from a lab. So you couldn't prove that it didn't come from a lab unless you have, as I say, able to prove that it has intermediate host. So simply showing that it's similar to a bat, so it doesn't show one way or another whether it came from bat or whether it came from a sample from bat in a laboratory. And the only way to determine that would be to sequence all of the samples that are present in all of the laboratories to determine whether those samples were actually present within any of those laboratories. So it's an open question right now. It's important to address because we need to know where it came from and not worrying about too many hurt feelings perhaps, and maybe not finger pointing too much, but to understand the true course of events is very important. So the evolution of SARS-CoV-2 during the pandemic, so it is, as I say, it is evolving, and it's actually evolving. There's already ancestor in December, November, China, and having tentatively called SNL. A number of other publications also describing essentially the same thing. L dominates in China, but outside of China, S has increased its frequency. This is very interesting. And the authors here, they propose that it's due to increased transmissibility of S. However, it could simply be what's called a founder effect, so drift. It could be a random effect. So essentially, the first seeding of the of SARS-CoV-2 from the China region into, say, Europe may have been of a different type, and so therefore that will then happen to dominate the population there, simply because it was first through the door. And so that's sort of what's called a neutral effect. So so future scenarios, how will the virus evolve? So the first scenario is that strict isolation testing and tracing may select against virulent strains and promote the evolution of benign strains. So this is a situation where human intervention might be altering the evolutionary trajectory of the pathogen. Actually, this happens all the time with drug interventions. And so now the second scenario is that strict quarantine may impede the acquisition of herd immunity, leading the population to be vulnerable to a second or multiple waves. And this is the current worry, of course. So a more lethal strain may emerge, and with an and it will be especially lethal if it has an asymptomatic transmission period, and then triggers a cytokine storm, as I say. And if the population doesn't have herd immunity, then it will be vulnerable, and herd immunity can be acquired by exposure to the pathogen itself, by vaccination. So the third scenario is that the virus will naturally evolve to become more benign, according to the trade-off hypothesis, as it adapts to the human host. However, given that transmission appears to occur before severe symptoms appear, there may actually be little selective to kill the host. So this may be a long, drawn-out process. So just to wrap up, I have an interest in evolution game theory, and it turns out game. And so we're engaged in a giant global signaling game with an emerging pathogen, and it's quite unprecedented, as I said, really because of the speed of spread of the pathogen, but also, as I'll show you in a minute, some of the strategies, counter strategies that we're utilizing, and some of the technologies that we're utilizing, all of which can be framed in terms of a signaling game. So a signaling game involves a sender and receiver, and so the sender sends a signal to the receiver, which undertakes an action when it receives that signal, and which produces utility, which is a benefit. Now, if there's common interest between sender and receiver, so in other words, if they're collaborators, they both receive utility when the receiver undertakes the action. Now, if there's a conflict of interest, then the sender will gain utility at the expense of the receiver. And so it's a cheat. And because in this scenario, the sender sends a deceptive signal to trick the receiver. And this can occur because due to something called information asymmetry. And so the sender possesses information regarding its true type. So only the sender really knows what it is. So the virus only knows what it is, whether it's a foe or friend. And, but it wants, in fact, it's a foe, of course. And it wants to obscure that from the host using all those strategies that I mentioned. And so, so only the sender knows whether it's a collaborator or a cheat. And so it's up to the receiver to try and determine the true type of the sender. And so obviously we have a conflict of interest with the virus. It wants our resources. So it wants to use our body, and it doesn't want to pay us for it. So it just wants to take stuff from us. And so it's a cheat, essentially. And so it's therefore obligated to use deception, because if we knew what it is and what it's up to, we wouldn't allow it to get away with what it's trying to do. So in general terms, the way the natural and man-made worlds are constructed is due to information asymmetry. This is the reason we have a legal system. This is the reason we have a Senate and a Capitolio, and we have debates, is to try and get to the truth. And the reason the truth isn't always apparent is due to information asymmetry. So, so the current pandemic is driven by information asymmetry between two players, the virus and humanity. And so whoever has control of the information will win the game. And so this is literally an info war. So we are on a war footing, but it's a war of information. And so, so it's info war, as I say, and This gentleman here is making a nuisance of himself recently. And by the way, I've never seen this program, but I've heard rumors. And he's actually engaged in forewarned, probably from the side of the deceiver, I would say, in terms of misinformation. But that's where it becomes interesting. And so, so just to wrap up, the sender, that is SARS CoV-2, utilizes a range of deceptive strategies. So it undergoes immunovation by using camouflage, so that is glycosylation, spike protein, replication organelles, molecular mimicry, such as mRNA capping that I mentioned, and of its own RNA to avoid detection, and many more. The better we understand the deceptive strategies, the better we can counter these deceptive strategies and combat these deceptive strategies. So understanding what's happening at the molecular level is very important for both priming our immune systems and also for designing drugs. Now, detection strategies of the receiver, which is humanity. And so, so at the organismal level and at the unconscious level, we are all born with an innate immune system and adaptive immune system. And their role is to detect the virus. And they do a pretty good job most of the time, but obviously in some folks it causes this cytokine storm, so it's not operating perfectly. Now, in addition, at the organismal level, we have vaccines and we have blood serum transfer, and these are consciously designed, and these utilize science. Now, at the societal level, we have all sorts of new and exciting testing kits to test for the presence of the virus. So again, this is reducing the information asymmetry, revealing its presence. And so one way we can do that is using the genome sequence, so using sequence-specific kits, and also using kits using antibodies. Unfortunately, these are not so accurate right now, but they have the advantage of being very quick. And so also social monitoring, and so observation of symptoms is one clue as to the presence of the virus, or the virus has revealed itself to us through its indirectly through the symptoms. Observation of outbreak clusters and contact tracing, and there's all sorts of sophisticated vigilance and our surveillance of all of these characteristics, which can allow us to detect the presence of the virus more efficiently. And so the bottom line is, the more information we have about the evasive strategies of the virus, the better. And the better we can detect the presence of the virus, the better, punto. And so obviously this comes from science, but it also comes from things like large-scale testing. So given that these tests are actually pretty cheap, there's no reason not to do large-scale testing, and it will help inform our public policy, no doubt. So that's it to finish up with. I'd just like to acknowledge my colleagues who've contributed to developing some of the ideas in this presentation. So Jose Carlos Rodriguez and Ash Maciek from Mayaguez, Tuguru Girai, who actually submitted me to write this initially, and then Miguel Urdaneta, who's kindly hosting this presentation, from Rio Piedras, Bud Mishra from NYU, and Will Casey from Naval Research Unit, who with whom I've been developing these ideas about mimicry and signaling game theory, and also to our host, Natural Sciences, Department of Biology in Rio Piedras, Puerto Rico Society of Microbiologists, and the Industrial Microbiology students. And so we can perhaps take some questions.
Yeah, hello.
Does anybody have any questions?
Yes, I have a question. I would like to know.
Hello.
Hi, my name is Valeria. I have a question.
Yeah.
I would like to know, will more virulent strains of the virus emerge?
Yes, so the answer is yes. So some will emerge, but whether they will spread is another question. So simply due to the random mutational process, some amino acid changes will produce more higher virulence in some viruses, individual virus genomes. The question is, that's an open question. It depends on the characteristics of the virus. As I say, if there's an extended asymptomatic period, then it may, given the widespread lockdowns, well, actually I should rephrase this. Given the widespread measures, some of the widespread measures that have been taken, that's likely to inhibit the spread of the virus, but perhaps not all of them. And I think we should be careful to assume that this sort of the peak and the drop in the peak is because we've been so strict with our quarantine procedures. I don't know that that's been proven yet. Certainly some of these things are working, but we're not sure as to, perhaps we can imagine. And I think the transmission rates have been shown to have dropped, but it's not clear whether simply covering your mouth and washing your hands is effective, and whether locking people up indoors all day is that effective or not. So, but given some of these measures, then it is inhibiting the spread of the virus, and so that would inhibit the spread of more pathogenic strains. However, a counter-adaptation, if you remember, I mentioned this arms race. So this is what we are doing, right? This is our sort of our strategic move is to implement all of these strategies. And so actually a counter move by the virus would be to extend this asymptomatic period. And so we might expect that to see that. And then, as I said, the virus is actually not causing the deaths. It's mostly it's actually the immune system is overreacting. And so I could envision a scenario where that might happen. But I think right now there's so much vigilance in health services around the world that we would hope that they would be vigilant to that occurrence. Does that answer your question? It's not a straightforward answer.
Yeah, it answers. Thank you.
Muy bien. Para organizar la discusión, si desean hacer una pregunta, pueden encender su micrófono o pueden también escribirlo en el chat, y así podemos ver las preguntas y contestarlas.
Okay, this is Tanok Henry. I just have a question. Is there any information that correlates the characteristic of the spike protein with the route of infectivity? There was a study published by the German research group about explaining that the infection starts with an upper respiratory attack, spends some time in the throat, and then migrates to the lung. So I was wondering if that's something to do with the molecule, with the spike molecule characteristics.
I'm sorry. Could you repeat that last bit? I couldn't hear the last bit.
So I was wondering if there's any correlation on the spike molecule characteristics and the route of infectivity and the preference for the cells in the throat, upper respiratory tract.
Yes, there is. Yes. So the spike, so the receptor binding domain of the spike protein is the conformation is complementary to that of the ACE2 protein, which is a human protein on the surface of cells, and so ACE2 is most commonly found in the respiratory tract. So it's on the surface of cells in the respiratory tract, and so this facilitates the entry of the SARS CoV-2 virus into cells of the respiratory tract, because essentially it's utilizing ACE2 in order to gain entry into those cells. And so the conformation of spike protein, the receptor binding domain of spike protein, is key to that interaction with ACE2. Now, there's some other coronaviruses. Not all coronaviruses bind to ACE2, so they do bind. They can have different receptor proteins. But in the case of SARS and SARS-CoV-2, they both bind to ACE2, which is most densely found within the respiratory tract.
Okay, thank you.
So, hi. Can you hear me?
Yes, hello.
So this question kind of links back
Go ahead, yes.
to the quarantine and the measures of the quarantine working or not. Do you think that SARS-CoV-2 will disappear from Puerto Rico completely?
That's a very good question. And this is where I think the signal game perspective is perhaps useful. And so what you're referring to, what's called a zero game, which is where humanity wins and the virus loses. So the virus just disappears. That rarely happens with pathogens. So we've not managed to basically completely remove most human pathogens. So they're out there. No matter what we do, we can't get rid of them. And so the analogy is more like between host and pathogen is more like an equilibrium that we come to basically an understanding. And essentially, the virus in the long term evolves to become less pathogenic, and so it affects the host less, and then the host is less bothered about the presence of the pathogen if it's causing less harm. And so they basically come to a compromise. And so in game theory, that's called a Nash equilibrium. And so we would expect an equilibrium between the two, and this is also the trade-off hypothesis proposes this as well. So the thought is that it's likely to become endemic, and it's likely to circulate within the population around the world, and this has happened with the Spanish flu. So the Spanish flu basically has become endemic, and it's still with us. So it's the descendants of Spanish flu are the flus that affect us sometimes during the winter. So that's seems to be the current prognosis. There's no reason to say viruses will do all sorts of things to maintain themselves within the population. Now, there is a proviso to that. We could extinguish the. I think I'm correct in saying that. And then, if you vaccinated the entire population around the world in one go, then yes, you could. If you didn't do that, then you would have reservoirs in different regions around the world, and it could still easily come back. But yes, so. But the good news is, over the long term, it's sort of expected to die down because we would expect it to adapt.
Yes, I have a question. Sorry. Yes, I have a question.
Hi.
My name is Fatia. Yes,
yes. Yeah, go ahead.
Does the COVID-19 have or could have something to do with the genetic edition technique, CRISPR?
Sorry, could you repeat that? Could it?
Does the COVID-19 have or could have something to do with the genetic edition technique, CRISPR?
Oh, okay. I have to make a confession. I'm not an expert on the CRISPR technique. I think the way that it's that you would sort of modify these small RNA genomes is just you're not editing necessarily. So CRISPR is for organismal genomes. And so with viruses, you actually purify the nucleic acid and you can synthesize them de novo. So I don't think the virus itself, one would use CRISPR for that. But as I say, I'm not an expert on CRISPR.
Thanks.
Hello.
Welcome.
Hello, can you hear me? Hello.
Hello.
Yes, my name is Maria. I was just.
Hello. Hi. Go ahead.
Yeah. I was just looking to the future scenarios about the virus evolving, and in the second scenario, you said about that the strict quarantine may impede in the acquisition of the herd immunity. So maybe we will be vulnerable for second or multiple waves. Do you think that if we are all like quarantined in our house would be a factor contributing for a future wave, because we won't be like creating a best habitat, or saying like that, to the virus to get like more similar to our RNA? Sorry. I believe that you said that once the virus was more available to the human genome, it will be like more similar to it, and it won't be so dangerous to us. And I'm asking you, at the second scenario that you told us. Right.
Yeah. So, right. So the second scenario was So by sort of trying to contain and inhibit, obviously that's a good thing because we're cutting the numbers of mortalities, but it has reducing herd immunity in the population. So it's reduced. If you reduce the total number of infections, then there's going to be less immunity in the population. Now, I have to stress that it's not clear how long the immunity lasts to SARS-CoV-2, and indeed if one can actually be reinfected with SARS-CoV-2. So that's not actually clear right now. But assuming that there's some level of protection from after you've become ill with SARS-CoV-2, then that acts as a barrier for the rest of the population. So the more people who have that immunity, they actually act as a shield, and they sort of inhibit the spread of that virus through the population. And so it has a beneficial effect. And so it's in itself, this is a trade-off between the two. And it's desirable to achieve herd immunity, but preferably through vaccination. Obviously,
okay, so
sort
it
of
will
run
be helpful.
sort of riot through the population is controversial. Yeah.
Yeah. So it will be helpful.
Sorry?
So it will be helpful. So if we have a lot of people that is asymptomatic, but is still infected, in sort of a way, it will be helpful because of the herd immunity.
I hadn't thought about that. Yeah, so I'm not an immunologist, so I don't know the extent of the immunity you would have if you're asymptomatic, because almost by definition, your immune system's not really recognizing the infection.
Yeah,
but
yeah.
So one sort of an alternative idea out there is that it is segmentation of the population, and so it is clear that younger folks really, the mortality rates are low, and so there are cases of mortality, but it's relatively very low compared to older folks, where it's very high. So there's very clear differentiation between those two segments. so one idea is to allow younger folks to go back to work, and then some of them will become infected through that process, unfortunately. But the objective shouldn't be to infect people ever. But that would sort of be a side effect, and they would actually act as a shield, because around the older folks, because that actually acts as a barrier to the older folks getting sick. So that's one idea that's out there. These are just ideas that people are throwing out there. There's no good option.
Okay, so basically,
these are difficult decisions.
So basically, the ones that survive the virus are the ones that will be helping us to create the herd immunity, right?
Yeah, and unfortunately, that's the way that it actually a very sort of transparent way of expressing it, because yeah, I mean it's an ethical dilemma, because you know, some this is actually called a trolley problem. You sacrifice, you know, knowingly put people in danger's way in order to save, you know, a larger segment of the population, and then actually the answer to that is no, and so often, and so you know, so we have to be very careful about how these things are approached, and but given that younger people, the mortality rates are very low, and you know, you can sort of make the argument that it's affecting people in other ways. You know, their mental well-being, their physical well-being, you know, etc. is also being affected. So, but it's a very clear differential. It's unusual with this pathogen that it's such a clear differential between very low mortality in folks under the age of 50, and not very high in folks over the age of 50.
Hi.
I like the idea of the passports the best, and there's still some flaws with that, but if you can actually test people, that would facilitate the process. Yeah.
Thank you.
Hi, Dr. Massey.
Hello. Yeah.
I'm Melissa from
Hello.
Ciencias Médicas. What do you think about sewage monitoring systems for the press? Do you hear me? Hi.
Yes. Yeah.
What do you think about sewage monitoring system for presence? I think. So, what do you think about sewage monitoring systems?
Sewage treatment?
No, sewage monitoring systems for the presence of SARS CoV-2.
Yeah. So my understanding is that it is excreted. So it's present in fecal matter. So it is secreted. I'm aware of a study in the Netherlands where from the Netherlands, where they were trying to monitor wastewater for the presence of the virus. I don't know what the results of that study were. I'm also told that there's a group in Washington State also with an interest in that. So there's a couple of people on the mainland with interest in that, I think. I understand, but yeah, I'm told that there's a publication or preprint by a Dutch group on that topic, but I don't know what the results. It's and this is you'll have to ask this of my colleague Jose Carlos Rodriguez, who works at the Botanical Gardens, because he's an expert on he would be able to answer that question for you better than me. And I can put you in touch with him if you would like.
Okay, thank you.
Okay, you're welcome.
Chief, there's another interesting questions in the chat. I don't know if you can see it.
Just a minute, I'm having a look. Yes. There's a lot of questions in the chat. Shall I? Oh wow, there's a lot of there's a lot of them. What shall I do? Shall I answer some of these questions?
Yeah, some of them. We are short on time, so but
Shall
you can
I have a go at
select some of
answering?
them. And yes.
Okay, so I'll start at the top then, shall I?
Okay.
Miguel? Yeah?
Yes.
Okay, so I'll
You can do it
start
yourself.
answering, unless anybody else has any sort of spoken. Yeah. Yeah, if anybody else has a spoken question. Does anybody have a...
Yeah, I have a question. My name is...
a verbal question?
Yes, I have a question. My name is Isaac Cortez.
Yeah.
Is it possible... Yeah.
Hello,
Dr. Massey. Is it possible that a person that was infected during the 2003 and 2004 pandemic of SARS CoV-1, is it possible that that person could be infected again with this COVID-19?
So I didn't hear the last bit of the question. You were talking about SARS CoV?
Yeah.
SARS-1?
Is it possible that the person that was infected and eventually survived the first pandemic in 2003 and 2004, is it possible that that person could become infected in this new pandemic?
Okay. Is there cross immunity? Again, well, there's two aspects to that. It's the level of similarity between SARS-1 and SARS-CoV-2, and they're actually quite similar to each other. And I forget the exact number, but it's something like 76% identical at nucleotide level. So they're pretty closely related, and so from that point of view, one might expect some cross immunity. The empirical studies, from what I recall, is that there is some cross immunity, but I forget the nature of that. So the other, and as I'm not an immunologist, but that is in the literature, I believe. So in other words, do antibodies to SARS recognize SARS-CoV-2? And I believe that there is some cross immunity. Now, whether a person who's had the SARS can also get SARS-CoV-2, you'd have to have the unusual situation where, you know, a survivor of SARS then is somehow sort of exposed to SARS-CoV-2, and the only way really is to see that is if they were to accidentally get infected, and then that would sort of answer, that wouldn't necessarily answer the question, because it could be that after 17 years after SARS-1, the immune system doesn't, immunity doesn't last for all that long, and it could have disappeared after that length of time. So I guess the answer is I'm not sure. There is a possibility of cross immunity though. Anybody have any more questions? Or spoken questions? So what I could do is some of you have sent some questions by the chat, and I could start to sort of go through those perhaps. And so the first one was Gary Taranzos asked, why is it that the phenomenon of the quasi-species in other RNA viruses is not seen with coronaviridae? It has been seen that there is this phenomenon with mares in dromedaries, which are camels, but apparently not in humans. This may be very important to determine virulence and transmissibility. in SARS-CoV-2. So, so the first thing to say about quasi-species is it really was to think about things such as retroviruses, such as HIV, which have very high mutation rate. And then they have this sort of, the idea is they have this swarm of mutants. And but really, I'm sort of, don't really adhere to this idea of quasi-species, because I think that it's, first of all, nobody can really define what it is. And secondly, yeah, it's true that the higher mutation rate, you're going to have a larger number of variants, but there's no such thing as species anyway in a virus. So so I'm a sort of skeptic regarding quasi-species. The MERS virus, which is the Middle Eastern Respiratory Syndrome, which is also another coronavirus. Actually, the ultimate origin of that was bats, and but I'm not so sure. I don't know about the mutational dynamics of that. And I agree with you that the mutation rate that you're referring to is very important for virulence and transmission. And then we had another question from Melissa Ortiz about the sewage monitoring, which I think we spoke about that one. And then we had another question about from Jacqueline Colon about will you get immunity, the people that survive virus SARS-CoV-2? And I think the answer, again, I'm not an immunologist, but I think the answer to that is that we actually don't know right now. It's unclear. Then Bez asked me a question about herd immunity, which I think we spoke about. Then we had a question from Rafael Tosaro. Strains are there in the S subtype? How much genetic? So that's a very good question. So I'm actually, there is a formality to describing something as a strain, and that's actually why I called it a subtype. So just to be to hedge my bets. And so really for strains, I think they need to be associated with phenotype, and they've not been associated with phenotypes yet. In addition, most of the mutations are actually what are called singletons, so they're not in juxtaposition with other mutations. At least that's my understanding. So how much genetic divergence is required to determine a new strain? That's a really and it sort of goes back to the species problem in biology as to what is a new species and what is the difference between a subspecies and a species. And it's not clear, but that's one for the virologists to make that sort of decision. And so there's a recent paper that's claiming that there's 3 strains, A, B, and C, and actually B is equivalent to L and S that I mentioned. And then there's a recent publication also on that's saying that there's 8 strains as well. So that's an important question of the syntax as to how do we define a strain. And we need to keep with the formality there. And so that's why right now I'm just calling them subtypes to be safe. Jose Augusto Rivera asked, is it known in China or any of the early countries if the frequency of mild or symptomatic cases is increasing? That's a very interesting question, and I don't know off the top of my head, but this is something that they would be looking for. So yes, researchers would be trying to associate positive tests with the symptomology. So and then but then of course controlling for other things such as age and health status and things like that. Marcos Delis is asking, why are some people asymptomatic? And that question I can't answer because as I say, I'm not. My knowledge of immunology is scandalously bad. So that I don't know. We should perhaps find an immunologist to address some of these questions. Camila Freitas. Where'd she go? Hominogues, if I pronounce that right, asks, no, SARS-CoV-2 is natural evolution or scientific development? Yes, very good question. I think we can't say that it's not a scientific development now. And at least that particular portion that I mentioned of the genome, there is a signature in there. We can't say, in my opinion, that is necessary due to natural evolution. And we can't either say that it's a scientific development. So right now, my sort of interpretation is that we don't have an answer to that question. There are no clear signatures that it's been engineered, or no unequivocal signatures that it's been engineered. Now, Maria Cortez Cobos asks, is there a risk factor of increased mortality in Puerto Rico due to the high number of people suffering from asthma? So that's a very good question, and that's another one for the immunologists, because I don't really understand asthma. It's an autoimmune disease, and so and it's an overreaction of the immune system. But I don't know if that would be a exacerbating factor in COVID-19. And the one thing that is interesting in Puerto Rico is that despite the low number of tests that we're having here, there is actually seem to be certainly mortality seems to be lower in Puerto Rico. Thank goodness. And so that is quite striking when you look at sort of temperate zones. And so there may be predisposing factors such as it's more of an aged population here perhaps, and as you mentioned, things like asthma, but actually the mortality is relatively low, still too high, but lower than elsewhere. And I say we're very lucky from that point of view that it could be worse. And I think it would be interesting to try and figure out why. Is it the very strict quarantine that we implemented very quickly? That could be a factor, or could it be other factors here in Puerto Rico? But Cobos is then saying, my question is because Puerto Rico mortality rates is higher. I think, so the way the mortality rates are commonly calculated are in terms of per positive tests, and it may be that it might look like it's higher in terms of the positive tests, but if you look at the size of the population and think of mortality that way, it's a bit lower than elsewhere. It's higher than some places, but it's actually lower than other places. New York being a good example, Italy obviously being a good example, Spain, United Kingdom, places like that. So it's in. I just did a quick sort of comparison with UK, which has had. It seems, well, they've had 17,000 recorded deaths, and it looks like it's 40% higher. So it's looking like something like 27,000 deaths, and it's 20 times bigger than Puerto Rico. So if you multiply the number of deaths in Puerto Rico, which is 65, I think is the current number, and then by 20, then we have 130, and we have 1,300 equivalents. So we're way lower than say the United Kingdom, and but it does depend on how you calculate it, and of course it is much higher than it should be. Perhaps we can make that argument, or we would like it to be a lot lower than it is.
Queremos agradecer al doctor Steven Massey por esta interesante conferencia. Hemos excedido el tiempo para darle la oportunidad a todos los que interesaban hacer preguntas y participar, pero ya el tiempo, ¿verdad?, se nos ha acabado. Queremos agradecer al doctor Steve Massey, también a los auspiciadores. Queremos agradecer a la Sociedad de Microbiólogos de Puerto Rico, que muchos de sus miembros participaron también en esta actividad. Obviamente a los estudiantes del curso de Microbiología Industrial, que son los que originaron esta actividad. Esperamos que la actividad les ayudara a tener un mayor entendimiento del tema. Y con esto nosotros concluimos la presentación de este seminario virtual. Y muchas gracias por participar y que tengan muy buenas tardes.